My name is Christopher Vakoc. I'm a cancer biologist at Cold Spring Harbor Laboratory. When I learned about DNA for the first time, there was nothing else I wanted to be in my life other than a biologist. I did a combined MD and PhD degree, and then afterwards decided to commit myself to basic research. My grant really has two major areas of focus. One is to develop strategies to target some of the most nefarious cancer-causing genes, in particular those that cause pediatric cancers. The second major focus is to develop a new tool to allow us to understand how drugs work. There is a major revolution occurring right now in biology, which is called the CRISPR craze. This technology is allowing biologists around the world to do things that we would never thought were possible. Our lab has made use of CRISPR to ask one very, very specific question, which is, can we discover new drug targets in cancer by applying CRISPR technologies, discover vulnerabilities that cancer cells have? We take a large number of candidate proteins in the cell, and we use CRISPR to sort of poke around at different surfaces on these large, complicated proteins. And we poke on a surface, and we basically ask the cancer cell, do you care? Does this affect you? Can you still grow and cause a tumor? And we can poke tens of thousands of surfaces on proteins in a systematic way. Discover these potential vulnerabilities, you could call them Achilles heels of a cancer cell. And we're basically exploring the potential effect a desirable drug might have. Essentially draw a bullseye around a compelling target. Specifically to this grant, the question that I considered was, what would make an impact in drug discovery? And the class of targets are something called fusion oncoproteins. Another way to describe that would be chimeras. And this chimera, this fusion, is the source of some of the most powerful cancer-causing agents that we know of. For that reason, we've decided to direct our CRISPR approach to fusion oncoproteins, and those cancers being among the most common in children. These targets are widely viewed as challenging or hard to make drugs against. And the reason is we don't actually have a visual of all of these proteins and what they look like. And we think this is the exact thing that our technique was built for. The second aspect of this project is to develop a tool that can be used in drug discovery to allow drug developers to demystify how the drug actually works. And so in this project, we're going to pursue a new technique that's going to allow a scientist, a chemist, a way to see the important target of a drug. The greatest compliment we could receive on our research is that we can watch pharma swoop in and take these things into patients.